Okay, I'm going to sketch a graph. And just like we did last week for temperature, if I plot on the x-axis, we've got pH. And here I've got rate of enzyme activity. We want to be brave. Give you a green pen. We want to be brave and sketch what they think might happen.
We've already done this, haven't we?
We did temperature.
Is that not? Oh, pH.
Yeah.
Would the same thing happen? Yeah.
Have some think about it. Do you want to go?
Try.
Go on then.
All I know is that it would be both. They'd be low on both sides because. I don't know.
Pretty good. Yeah, pretty good. I'll just tidy it up. That's pretty good. Well done, Ray. Thank you very much. Okay, so what we find is that we would have an optimum pH. Okay, an optimum pH. Okay, and then either side we have decreased activity with decreased activity either side. Of that pH. Okay, should put here NB. I'm going to really want to stress this because it didn't seem to get through to my current grade 12s. So I really want you to pay attention to this. Different enzymes will have different optimum pHs. Sorry, that pen is really blue. Yeah. Different optimum pH is and temperatures. Okay, I want to just... So different enzymes have different optimum pHs, okay? What different pHs do you have in your body, in the human body? Do you know?
I assume that there's a different pH inside your stomach and inside your organs than outside of them.
Yeah, so there's, your stomach has a pH of around 2. Very, very acidic. Okay. Most of your other tissues are around pH 7. All right. There are enzymes that are optimized to work in your stomach. Okay, their optimum pH is around 2 or 3. Okay, they work best at that pH. Other enzymes that work in other environments in your body have a different optimum pH. Okay, so enzymes tend to be, you tend to find whatever environment they work in, that is their optimum environment to work in. Okay? With temperature, different organisms, particularly bacteria, right, live at extremely different temperatures. Okay? and they might even use similar enzymes to us. So, for example, amylase is an enzyme, right? Is optimized. Human amylase is optimized to work at human body temperature, 37.5 degrees. Okay, but that's not necessarily the same for bacterial amylase. Bacterial amylase that might from a bacteria that lives in a very hot environment, like you get in hot springs. You get bacteria that live in those springs as well. Okay, and extreme examples are actually boiling water. You get some bacteria live in water that's 80 degrees C. Okay, their enzymes are optimized to work at that temperature. Okay, that's really important just to be aware of that. Don't just blanket assume that every enzyme works at 37 degrees centigrade. Okay, it's not quite true. Okay, all right. So what's happening here? What's happening to the enzyme either side of the optimum? Any suggestion? Why is the activity decreasing, do you think?
Because the pH number is getting higher.
So the pH has changed, or the pH number's gone down or up, yeah? But what's actually happening to the enzyme means that it's no longer functioning.
Is it the chemical?
It's to do with the chemistry. Okay, so here we have the enzyme is denatured. Okay, so the enzyme becomes denatured here and here. Okay.
In the middle of the. So not at the top.
So the optimum the optimum the enzyme works perfectly normal, but either side of that optimum the enzyme becomes denatured. Okay, and it's to do with the change in the chemical properties of the active site. And we're not going to get involved. None of you are chemists. We're not going to get drilled down into the actual chemistry of why that happens. But you have to just take it from me, I'm afraid. Okay, you have to just accept what I tell you is the truth. All right, but basically, when the pH changes, that affects the R groups of the amino acids in the active site, changes their properties. Okay, so this changes the chemical properties of the active site, okay, which meaning the enzyme no longer works. All right, not enough coffee this morning.
It's just really cold out.
I know, it's freezing. I had fun driving into work today. It's like an ice rink.
Our trains are delayed, so we
Yeah.
need to sit there for like
Oh.
15 minutes in the cold.
Oh gosh.
People
Yeah.
look like
Minus
penguins.
-8, yeah.
Yeah.
Warm up.
Stay outside with -8 extra.
Minus
Yeah.
12
Yeah.
temperatures.
Oh, look at this. Typical. It's like, oh, yeah, -12. Hot stuff. Yeah. All right. Okay, can I rub this off? Move on. So the last thing is substrate concentration. We'll just talk about substrate concentration. Okay. You need to have your brain switched on for this one. It's a bit of a buck the trend slightly. So, what do I mean by substrate concentration?
How much of the substrate is present?
Yeah, so per volume, unit of volume, it's the number of substrate molecules in that unit of volume. So whether you measure centimeter cubed or meters cubed or millimeters cubed or micrometers cubed, the concentration is a count of how many molecules are actually in that fixed space. Okay? So higher concentration, you have more of those substrate molecules per cubic measurement. Okay? Okay, and we can represent concentration with square brackets. It's just a shorthand, it means concentration. So if I write substrate and put it in square brackets, that just means substrate concentration. You might see me do that sometimes. Okay, it's just sort of a shorthand. Okay, so as we increase the concentration of the substrate, what do you think might happen?
More reactions. There's more chance of a reaction happening.
Why?
between the enzymes because there's more substrates.
Yep.
So there's more collisions.
Yeah, there's more chance of those molecules colliding. Yeah. Okay, just like if there's more cars on a motorway, there is a higher chance of a collision between those cars, right? In heavy traffic, makes driving a bit more stressful. Okay, just remember your wits about you a bit more.
The effect of substrate concentration on the rate of.
This is the the effect of, thank you, Liz, substrate on enzyme activity. Okay, what do you think that increase is going to look like? Is it going to be a linear increase? Is it going to be an exponential increase? Is it going to plateau? So, a linear increase would look like a straight line. So A, that's one option. An exponential increase would look like, yep, like a little like a half pipe, quarter pipe, whatever you call it. Yep. Okay, so B, or might it do this? C. Okay, let's vote. Hands up for A. Or do you want some thinking time? I'll give you some thinking time. Give you a minute to think about it. Raphael, which one are you going for?
It depends on the concentration, right?
So as the concentration increases, if you were to
Okay,
plot
I see. So
concentration increasing with the rate of activity, would you expect the rate to increase at the same proportion? Would you expect, as the substrate increases, not much increase initially, and then it suddenly the increase gets faster?
I think it's B.
Or we'd have lots of increase straight away, and then it might slow down?
B.
You think B? Okay. Why? I'm going to always ask for a justification.
Because it will start, so like the concentration will get with time will get like more concentrated.
So as you get
So
more
it's
and more
not
concentrated, you think it gets more and more and more and more
So it
faster.
gets- yeah.
Okay, cool. All right. Leo?
I think it's B or C.
You think B or C? Okay. Got to go with one. You're a betting man. Come on. C. Right. Why?
Because when there is more substrates, it's going to speed up the reaction. So
What's happening here as we add more substrate? What's happening?
it's going to decrease when it's going to get optimum.
Okay, but it's staying flat. This is showing you as what's happened here is it's increased a lot, but then as you add more and more substrate, you increase even further. There's no change to the rate. That's that flat line. There's no change. Staying at the same rate. As we increase more and more and more substrate, this is staying at the same rate.
But when at the moment there is going to be too many substrates, so that it's not going to affect something.
Okay. Okay.
Changing.
Okay. Cool. All right. So you're going to stick with C, huh? Good man. Right. Rare.
C.
Why?
Because at some point there'll be too many present, and I think there'll be too many collisions for it to work with all these enzymes. Because no one's increasing the rate of the amount of enzymes.
Okay, you're on the right lines. Okay. Yeah.
Well, I think I like B, but I think it has to be C because it cannot go on forever.
Okay.
It just it
I
can't.
think that's right.
It has to plateau at some point.
Okay. All right.
I'm gonna go with C.
All right.
Because like I think the more substrate concentration like increases, But it will get to a certain point where no matter how high the concentration goes, it's not going to change anything.
Okay, so you're right. The answer is actually C. Okay, the answer is C. Okay, why? We're now going to try and explain this. So we have this. Let's just describe this graph. I'll draw it a bit more neatly. Let's describe this graph. Just don't explain it. Just describe what you see. Remember we talked about this last week. Description, literally referring to the axes labels.
A substrate concentration increase.
So this is for 2 marks. 2 marks.
The rate of enzyme activity will increase until it reaches the optimum where it will plateau. Where it plateaus, not where it will plateau.
Okay, all right. So yeah. So 2 marks. Notice the division here. The best way to say this is at low substrate concentrations. So at low substrate concentrations, increasing the concentration increases the rate of activity. At higher substrate concentrations, increasing the substrate concentration has no effect. Okay, so when you come to your internal investigation, your individual investigation, and in the analysis section, you've got to describe your results. This is what you're meant to do. Okay, look at the trends you have and describe it, and then in your evaluation, you've got to explain it with the science. Okay, so at low substrate concentrations, increasing concentration increases the rate of reaction. Okay, see my shorthand there? So when you go to university and you're sitting in a lecture, you've got to develop a way of actually writing stuff fast, you know, that you can then expand on later. Yeah. So at high substrate, okay, increasing substrate concentration has no effect. And why? Why do you think? What could be a possible reason, way to explain this phenomenon, this pattern that we see?
Before the optimum substrate
Yep.
concentration, by increasing the substrate concentration, you're actually increasing the chances of collisions happening
Yep.
between the enzyme and the substrate.
Yeah, perfect.
But when you get to the optimum, it gets to the point where it's too many. I think this, no, because
Something becomes limiting.
the amount of enzymes. Like,
Yes,
so you're
and
not
affects
increasing
the amount of active sites.
Okay.
Okay? Because at high substrate concentrations, okay, the number of available active sites okay, limits the reaction. What does that mean? It means if you've got 100 enzymes, at some point, all of those active sites are filled with substrates and are working at their top, their fastest speed. And you can keep adding more and more and more substrate, and it's not gonna have an effect because those active sites are all filled. To speed up the reaction, you have to then increase the concentration of the enzyme itself. Okay? Any questions about that? I'm going to stop that there.